I hope by now you're familiar with the basics of cellular respiration, which is the complete breakdown of substrates like glucose within the cells to yield energy in the form of ATP. In this video, we're going to talk about the final step in aerobic respiration, which involves the electron transport chain. Before we go ahead and talk about how this is the final step and what is produced as a result, let's take a brief look at what happens before this electron transport chain can take place. So we start off the cellular respiration in the cytoplasm with glucose. Glucose is converted into pyruvate, which is a 3-carbon molecule, by the process of glycolysis. Pyruvate is then moved to the matrix of the mitochondria, where it is converted to a 2-carbon compound known as acetyl coenzyme A or acetyl-CoA. A carbon dioxide is released as a byproduct during this reaction. In the matrix itself, this acetyl-CoA enters the citric acid cycle, as a result of which we get two important molecules, NADH and FADH2. NADH stands for nicotinamide, adenine, dinucleotide, hydrogen. FADH2 stands for flavin, adenine, dinucleotide, hydrogen. So what are these two, NADH and FADH2? These are coenzymes that are going to act as our electron carriers. They are going to participate in this electron transport chain and finally give a large yield of ATP. That is why they are produced in the citric acid cycle, because they act as electron carriers for this electron transport chain. So from this, can you guess where this electron transport chain will take place? Because the products that we're interested in, NADH and FADH2, they are produced in the matrix of the mitochondria. From this, you may be able to guess that the final reaction, this electron transport chain, and the final production of ATP takes place in the mitochondria. And this is where finally we're going to use oxygen. Oxygen is going to be used up in this reaction, which is why aerobic respiration needs oxygen to take place, which is why a lot of organisms, they breathe in oxygen so that they can completely oxidize this glucose to yield ATP. Let's take a closer look at the mitochondria to understand where this electron transport chain is going to take place. So mitochondria, as you might be aware of, is a double membraned structure. So this is the outer membrane. The inner membrane is not straight like the outer membrane. It is made up of several infoldings like this, known as cristae. And the cristae has these infoldings to increase the surface area for the electron transport chain. So the membranes, as you know, are made up of the phospholipid bilayer. That's why I have drawn them like this. This is the phospholipid bilayer. This is the outer membrane and this is the inner membrane. And this space right here between the two membranes, this is known as the intermembrane space. And this, of course, is inside the mitochondria, which is the matrix. So this is how the mitochondria looks like, and we are very interested in the matrix because we know this is where NADH and FADH2 have been produced. But more than that, we're going to be seriously interested in this inner membrane of the mitochondria, this membrane that has these infoldings, because this is where the protein complexes that make up the electron transport chain are embedded. So this is how the electron transport chain looks like. You have 4 protein complexes, protein complex 1, 2, 3 and 4. You also have these smaller complexes known as the coenzyme Q and cytochrome C. So these protein complexes, they form the electron transport chain. There is another very important molecule that is embedded within this inner membrane, that is ATP synthase. We'll come to this in just a while. So now we'll take a look at the process of this electron transport chain, how it's going to take place. So we have NADH and FADH2 produced in the matrix of the mitochondria as a result of citric acid cycle. So what's going to happen is these electron carriers are going to come and donate their electrons to these complexes. NADH is going to come and give electrons to this complex 1. So this complex does not keep the electrons to itself. What it does is it transfers the electron or transports it to this coenzyme Q right here. And when this transport of electrons is taking place, energy is released. And with the release of this energy, this is used to transport protons that are in the matrix to the intermembrane space. We call this the pumping of protons. So with the energy that is released by the transport of electrons from one complex to the other, protons are pumped from the matrix into the intermembrane space. So the concentration of the protons is going to keep increasing in the intermembrane space while it's going to decrease within the matrix. So this is complex 1, which receives electrons from NADH. FADH2 transports the electrons to complex 2, from where the electrons are transported to coenzyme Q. The electrons are then transported from coenzyme Q to complex 3. When again there is a corresponding pumping of protons from the matrix into the intermembrane space, and then from complex 3 to cytochrome C, again protons are pumped. You might be wondering why these protons are being pumped. Well, there is a reason for that. Before we get to that, let's see what happens when the electrons finally reach this complex 4. This complex 4 is very important because this is where oxygen is located. And that is why oxygen is known as the final electron acceptor in aerobic reactions, aerobic respiration. It receives the electrons from complex 4 and is converted into water, which is released as a byproduct. So with the transfer of electrons to oxygen here, the electron transport chain is completed. We have all the protons pumped from the matrix to the intermembrane space. So now let me answer the question, why are these protons pumped into the intermembrane space? That has to do with ATP synthase. You see, ATP synthase is the enzyme, is the protein complex that's going to convert ADP and inorganic phosphate to give ATP. And for this ATP synthase to work, it needs the help of these protons. ATP synthase, if you remember from the video about light reaction, is like a turbine. It has a turbine-like component, which is this part right here at the matrix. So what happens is the protons, they flow back into the matrix through this ATP synthase. And when that is happening, the turbine component of ATP synthase, it turns or it spins around, which brings the ADP and inorganic phosphate close to one another, forming ATP. That is why protons are pumped into the intermembrane space so that they can be brought back into the matrix through ATP synthase, and the energy that is produced by the transport of these protons is used to convert or to catalyze the reaction between adenosine diphosphate and inorganic phosphate to give adenosine triphosphate. So this is the reason why this entire electron transport chain also takes place, so that by transporting electrons through the different complexes, we can correspondingly pump protons into the intermembrane space and then use this proton motive force to produce ATP. And because it uses or it sort of couples the reaction between this proton motive force and the use of oxygen, this entire process by which ATP is produced is known as oxidative phosphorylation. And this is all about the electron transport chain, at the end of which approximately 38 molecules of ATP are produced from just one glucose molecule. So you can just imagine how many glucose molecules will make up a food like bread, and from that one slice of bread, how many ATP molecules can be produced. That is a mind-boggling number.